Good day, everyone. I am Mark Angelo Argalendes, and we will be discussing our ninth topic for histopathologic and cytologic techniques laboratory, and our second discussion for the finals term, which will be all about staining using the hematoxylin and eosin method. So, why should we stain? Because simply, unstained tissue lacks contrast. All of the fixed material have the same or similar refractive index and similar color. So you would see a uniform dull, gray, or even brown color. As you can see here, ito yung itsura ng unstained tissue. 'Di ba? You cannot appreciate the morphology, the cells, the nucleus, the internal infrastructures of the cell as opposed to a stained tissue. So, using dyes to distinguish different cell parts is the reason why we perform staining, to better emphasize the architecture, the differences in histology, and even at the cellular level. The staining method of choice for routine histopathologic specimen is hematoxylin and eosin. It is used for routine microanatomical examination, of which the stains were introduced by Boehmer on 1865 and Fisher on 1875. So it is often used in tissue staining by Wisowski of 1876. So again, this is the appearance of an unstained tissue specimen. However, using hematoxylin and eosin, you will find out later that there are some structures that appear blue or purple, while some structures would appear pink to red in appearance. So the principle of H&E is banking on the dyes exploiting differences in tissue chemistry. Kasi 'di ba yung mga tissues natin, some of them, they have a characteristic acidic, basophilic, or even ionic na characteristics. So, it's primarily banking on ionic bonding, wherein electrostatic attraction of fixed tissue and dye causes the tissue to hold on to the dye. That is why it gains the color of the dye. Pero that's to make things simple. Let's first look into the primary stain hematoxylin. It is technically not a dye because it does not directly stain the tissue. In order for the tissue to gain the color of the hematoxylin, it makes use of a mordant, which is usually a metallic cation such as alum or aluminum, and later you will find out na meron pa palang ibang mordant. So the hematoxylin stain is actually comprised of the hematoxylin plus aluminum salts, which gives it its cationic or basic property. So this cationic basic dye will stain acidic components or anionic. And these tissue structures that are stained by cationic or basic dyes are what we refer to as basophilic. So what's an example? Look at the image here. As you can see in the nucleus, they're all darkly staining, mean to say they are stained by hematoxylin. Remember that hematoxylin is basic. Ano ba yung meron sa nucleus natin? Nucleic acid. So, because it is nucleic acid, it is acidic, which will then have affinity to basic and cationic dyes. Therefore, the nucleus and nucleic acid are basophilic because they are acidic components which take up cationic and basic dyes such as hematoxylin. So here are variations of hematoxylin according to the mordants being employed. So before we move on to mordants, I want you to have a better understanding on what mordants are. Okay. So let's say for example, this is your hematoxylin. Hematoxylin would not bind with tissue. Let's say that this is your tissue. Bakit hindi siya mag-bind? Because the hematoxylin would require mordants in order to facilitate the binding of the stain to the tissue. So, how does a mordant work? The mordant acts as a bridge. So, this is what a mordant does. The stain will now bind to the mordant, and the mordant will now bind to the tissue. meaning to say, the mordant is required in order for the staining to proceed. Now, do not be confused with the term accentuators, because accentuators are similar to mordants in the sense that they proceed the staining reaction. However, the staining can occur even in the absence of accentuators. So what does it look like? So this is your stain, and this is your tissue. Now, with the presence of accentuators, it would actually make the reaction faster. But the stain does not need the accentuator in order for the reaction to proceed. Again, this is the accentuator. It makes the reaction faster, allowing the tissue to take up the stain faster. But in the absence of accentuators, the reaction of the stain and the tissue can still proceed, as opposed to mordants, wherein the absence of a mordants, there will be no formation of the tissue dye lake complex, meaning the staining will not proceed. Okay, knowing na yun sila, let's go back to our mordants. So for hematoxylin, you have typically 3 different types of mordant. You have your aluminum or the alum salts, which gives the color blue of hematoxylin. The examples include the Ehrlichs, Harris, Coles, and Mayers, all of which are something that you have to memorize because they are typically being asked in the board examination. So they could be used for progressive or regressive staining. So just a little bit of review, ano ba yung progressive and regressive staining? Pag progressive, typically we just add a dye, add another dye, and then that's it. For regressive naman, we add a dye, we add a decolorizer, then we add another dye. Yun yung regressive. Pabaliktad. Okay. Now let's move on to iron mordants or the ferric salt mordants, which gives the characteristic blue-black appearance. Examples of which includes the Weigert's and the Heidenhain's hematoxylin, which is typically employed in regressive staining only. You also have your aqueous phosphotungstic acid. So, yung 1% aqueous phosphotungstic acid, isa lang yung ginagamitan natin yan. Or, yun yung tinatawag natin na solution na pita. Or, the phosphotungstic acid hematoxylin, which is usually employed for progressive staining. Again, all of the various types of hematoxylin are something that you have to be able to remember by heart. Okay? Now, let's move on to the secondary stain, which is eosin, and it is an anionic or acid dye. It reacts with cations which are acidophilic. So, ganito yun siya. Look at the image here. So, this image is what we refer to as an eosinophilic structure. Bakit siya eosinophilic? Because it has a high affinity for eosin. Ano ba yung mataas sa cytoplasm? Proteins. And some proteins are very rich in amino acids which are typically basic. Because it is basic, then it would have an affinity to acidic or anionic dyes. Kaya, it would have an eosinophilic appearance, and those structures are also acidophilic because they are basic. They love acid. Acidophilic. Examples of Eosin Y stains include the Eosin Y, which is yellowish, Eosin B or erythrosin B, then you also have your ethyl S. Both of Eosin B and ethyl S are rarely used. And Eosin Y naman is siya yung commonly used. Okay? So here are the basic steps associated with staining and mounting. First is deparaffinization, hydration, removal of mercury pigments, staining, dehydration, and clearing, of which dadaanan natin siya ngayon. So deparaffinization, as its name suggests, it involves complete removal of wax with xylene. So it requires 2 to 3 changes of xylene, and tissues should appear clear and transparent, and there should be no wax patches. So deparaffinization can be performed with xylene, or it could also be used using a hot oven, simply just by melting off the paraffin from the slide. Next is hydration. With removal of the wax and infiltration with xylene, remember that xylene is a clearing agent. It is immiscible with stains. So, we need to replace the xylene from the tissue with water. So, if dehydration requires ascending grades of alcohol, rehydration requires descending grades of alcohol, and the last portion is distilled water. So the presence of clear areas indicative of xylene. So you should rehydrate. Then removal of mercury pigments if necessary, especially if the fixative agent that you use is mercury. So tissues are treated with iodine solutions, of which, as mercury pigments react with iodine, then there will be the formation of tetrathionate with the addition of thiosulfate. So tetrathionate now is soluble in water, and we wash the tissue in running water to remove tetrathionate. Then removal of mercury pigments if necessary, especially if the fixative agent that you use is mercury. So tissues are treated with iodine solutions, of which, as mercury pigments react with iodine, then there will be the formation of tetrathionate with the addition of thiosulfate. So tetrathionate now is soluble in water, and we wash the tissue in running water to remove tetrathionate. Okay, so next you have your staining. So for the staining procedure, please just refer to the laboratory manual, but in general, it involves first is hematoxylin, then acid alcohol, and ammonia water. Now, I want you to take note that ammonia water is a bluing agent. And as its name suggests, it is a bluing agent because it renders the stained tissue with hematoxylin blue. But other than that, the ammonia water would also allow the eosin Y to properly bind with the tissue. After staining, then you should perform dehydration and clearing. So, just go back to the procedure for dehydration wherein you would use increasing concentrations of alcohol, and then the problem is that alcohol can remove stains, so kailangan i-modify mo to reduce contact time to minimize fading. And dehydrated tissues is then cleared with 2 to 3 changes of xylene. And remember that xylene turns milky when there is the presence of water in cases of incomplete dehydration. And that covers staining using the routine hematoxylin and eosin. Thank you for listening.